So at this time, I'll declare the annual meeting of stockholders to be officially adjourned. The business portion of our program for the day has concluded. All stockholders are invited to stay on for a corporate update presentation. I'd like to thank each of you as stockholders who sent in a proxy for this meeting, and I'd especially like to thank you who are here for virtually attending today's meeting and for your continuing support of the company. And between meetings, please don't hesitate to communicate with us. We enjoy interacting with all of you. Now, if you go to the website on the left, you'll see on the top two icons, the letter I, and to the right of it, a document icon. The document icon is the corporate presentation, which I'll quickly walk you through, an update on the company. It's titled Annual Meeting of Stockholders 2020. You'll need to advance the slides yourself. I'll tell you what slide we're viewing as I walk you through them. Slide number 2 is the forward-looking statements, and so I'll need to remind you that I'll be making certain forward-looking statements in this update that have associated risks and uncertainties, and so we refer you to our filings with the Securities and Exchange Commission for more detailed information about our company. Slide 3 is a recap of our mission. As the corporate name implies, we're focused on targeting the largest single and growing markets in medicine. Those are age-related degenerative disease. And we're employing in this product development program advanced technologies that can, we believe, reverse the aging of cells and induce tissue regeneration in diverse parts of the body. I use that term reverse aging very measuredly. I'm a gerontologist, spent my entire adult career in the field of gerontology, and I find it a remarkable thing that I can say here at the close of the year 2020 that technology is in hand to reverse the aging of human cells for therapeutic effect. It's certainly a remarkable time, a remarkable technology, and it's remarkable that we as a company have such a strong proprietary position in this technology. Slide 4 you may have seen before. This is the tsunami of aging hitting our shores. This year, I think we're all tired of hearing about COVID-19. But here was an epidemic that came to our shores. An even greater tsunami is not being addressed in the news media, and it's a tsunami of aging, and it will lead to even more serious health consequences to the United States. You can see at the year 2020, this inflection point is really beginning. When the number of people greater than 80 years old really begins to hit the clinic. And this carries with it enormous strains on our benefits programs and entitlement programs like Medicare, our ability as a country to properly care for the chronic age-related degenerative diseases of our population in the United States. And of course, this is true around the world as well. Slide 5 just puts in perspective some of the most powerful technologies in the history of biotech are platform technologies that allow us to manufacture to scale products that are previously rare but valuable for therapy. The biotech industry began with recombinant DNA, which allowed us to manufacture to scale recombinant protein. That's laboratory-produced proteins like insulin and growth hormone. In the old days, we had to squeeze insulin out of the pancreas of pigs. Now we can make it in bioreactors and companies with dramatically improved quality and cost of goods. The next revolution, similar, is the monoclonal antibody technology. Of course, this is widely used now in medicine. It allows us to manufacture to scale, again, highly qualified, high-quality, purified antibody, which can recognize specific pathogens like the coronavirus, the novel coronavirus, but also can be used to transport as a silver bullet agents to cancer and many other applications in medicine. The revolution in which I was involved with is regenerative medicine. This began with the isolation of pluripotent stem cells that I was proud to be associated with some years ago, and is now leading to billion-dollar acquisitions and an inflection point in itself, the new type of regenerative medicine that I think will end up being the most valuable, which we refer to as ITR, or Induced Tissue Regeneration, which I'll address in today's presentation. Well, back to cell-based therapy. So there's a lot of confusion in I think the marketplace about stem cells. The most powerful stem cell, or the one that is the basis of AgeX, is the pluripotent stem cell. These are cells that are capable of really dramatic, I can't emphasize enough how dramatic advances in cell-based therapeutic manufacturing. First, they're pluripotent. They allow us to make any of the cell types of the human body, literally everything. If it's a pigmented epithelial cell in the retina that causes macular degeneration, you can make those. If it's a cell for Parkinson's disease, you could make those. It allows you to make precise genetic modifications in cells, which has really generated a lot of value in recent years, for instance, with CAR T technology, which many of you may have heard of. And for us, it makes possible the genetic modification of any cell type to make them off-the-shelf transplantable. As many of you may know, cells can be rejected by the body as foreign, and we have a technology which we believe will mask immune rejection, which I'll be telling you about. All of this is enabled by pluripotent stem cell technology, really our cornerstone. Slide 7 drills down on some of the specific technologies that make us unique, our special sauce, so to speak. PureStem is a major technology in that regard, which we are a leader in. Slide 8 shows the significance of PureStem. On the top of this picture, you'll see traditional manufacture. The vast majority, if not all, of our competition are using pluripotent cells, either IPS or ES cells, and then differentiating them in the laboratory to make product. But as I'm showing you in this diagram, we have a problem of impurities. If we were making a drug and we had impurities, it's not the end of the world. Virtually every pill that you might take from the pharmacist's shelf has some degree of impurities, but your body can deal with those. And with living cells, an impurity can be a problem. These cells are alive, and they could make an inappropriate type of tissue. So, for instance, if you had cells injected into your heart, theoretically, for heart failure, and there were cells from some other type of bone cells, for instance, you would grow, a bone in your heart muscle. That would not be desirable. PureStem is a novel manufacturing technology which scientists in our company invented, and it allows us to make single cell derived, clonal, as we say, clonally purified or monoclonal lineages of cells, which are purified and directly scalable, dramatically advancing the ease of manufacture, and we believe ultimately FDA approval of such therapeutics. Slide 9 shows another important technology we bring to the table, UniverCyte. This is our masking technology to prevent immune rejection. It's detailed a bit more on slide 10. So HLA-G is a natural protein. By the way, the HLA genes are the genes that you try to match up. If you have a kidney transplant, you try to match up the HLA genes to match yours. That's when you're looking for a donor. That's what doctors use to try to align the kidney to you to prevent immune rejection. HLA-G is a type of HLA that nature uses to mask the developing pregnancy from rejection by the mother. As many of you may know, a woman can carry a pregnancy that has no genetic relationship to her, and yet that is not rejected by the body as foreign, and in part that's due to the masking of the pregnancy by HLA-G. So it's a natural protein, and we believe that natural aspect, it makes it highly desirable, and it appears to mask multiple arms of the immune system, as we're showing you in this diagram. So nature has evolved numerous different ways of attacking foreign agents in the body, and HLA-G acts against multiple aspects of immune response. So what we're developing are universal, or we call them UniverCyte cells. They're pluripotent that can be used to make off-the-shelf products for all patients without transplant rejection. And in that respect, these products are not unlike standard biologicals. Product that's manufactured at a centralized facility. Product is distributed as frozen inventory, as we're seeing with the vaccines currently being distributed, although they would not have to necessarily be transported in the same manner, and then stored at the point of care for administration to patients. Lastly, the company has HiStem as a matrix for the delivery of cells. Simply injecting cells into many tissues in the body is not desirable. The cells need to be sort of fixed in place with a matrix, sort of like bricks are put into a wall with mortar. Cells need to be engrafted in a solid matrix in the body, and HiStem has been licensed to us for the purposes of our lead product development, which I'm going to tell you about next. Business strategy, you've heard us speak a lot about the bailment of our master cell banks. I want to quickly walk you through why that's, we think, is a fantastic strategy. Slide 14 briefly describes this. So the master cell banks are the cells that are pluripotent cells that are stored under GMP conditions and could be made into various kinds of cell types for transplantation under GMP conditions as someday clinically approved products. And AgeX, under these bailments, would retain ownership of the master cell banks, that is the pluripotent cells. Our licensees would own the derivative products, but for defined fields of use. So let me just clarify this. So we would essentially rent them or bail, under a bailment agreement, we'd rent them the master cell banks. We have done this and we'll be continuing to do this. And we'll retain ownership of the master cell banks. It'll be our property. But the licensee will be able to manufacture, let's say, theoretically a heart muscle cell, and they would then own and could sell the heart muscle cells while we retain the ownership of the master cell bank, which would be required for the continual manufacture of the product. In exchange for the bailment, we'll receive a royalty. Again, this is not a patent license that can expire. It's a rental of the master cell bank, and so it would run as long as they use that cell bank. That results in a higher net present value to our shareholders than for instance, a patent license would. which typically, patents don't last as long as we would all like them to. Slide 15 is a summary of where we're really trying to maximize value creation in all the diverse applications of pluripotent cells. In the middle there you see a colony of these pluripotent cells, unformed cells. And of course they can be turned into all these diverse human cellular therapeutics. And the numbers and diversity of the types of cells that are being developed are growing every year. They can also be used for drug discovery, I'll touch upon briefly, to manufacture biomaterials like extracellular proteins and growth factors and things that cells make. And they can also be used for ectopic organoids. And this is a field that has growing interest in the regenerative medicine field. The generation of primitive tissues outside of the body for both research, drug discovery, and for transplantation. Slide 16.
Growing interest.
Partnerships in these non-core applications would be for the therapeutic use of the organoids, diverse biomaterials, including exosomes. So these are packages of messenger RNA that are secreted out of the cells, and adult cardiac cells, which is a proprietary technology we developed, which I'll be telling you about next. Slide 17 is from GE Healthcare, now called Cytiva. And they're here in this slide reminding us of many drugs that have been, even in many cases, approved for therapeutic use, and then what were removed from the market because of unanticipated cardiotoxicity. This is a major problem for the drug industry. How do we solve this problem? And we think we have an important solution to this. So drugs are typically tested in animal models to determine whether they're toxic to heart, causing arrhythmias or heart failure or other damage to the heart. Unfortunately, these animal models don't always pick up the toxicity. Humans are not mice. We're different. And so the ability to make human heart muscle for drug screening was picked up on the early days when it was then GE Healthcare, and we formed a collaboration with them. And what we have developed since then is a way to fully mature these heart muscle cells to be adult-like human heart muscle cells. And we believe this new development by our company will have significant commercial applications in drug screening, and we plan to move that forward in the coming months and years. Slide 18, I'll quickly walk you through our two lead programs, AgeX BAT1 and VASC1. Slide 19 reminds us of what brown adipocytes are. We have, in broad strokes, two different kinds of fat in the body. We've all heard about white fat. That's the type that we try to get rid of, especially around the new year. And we can do that by maintaining this balance I'm showing you here in slide 19 by controlling energy intake, not eating as many calories. The third axis to this metabolism, which is regulated by another kind of fat called brown fat, or brown adipose tissue, or BAT, in this diagram. What is brown fat? Well, in certain parts of the body, we have reservoirs of brown fat. On slide 20, I'm showing you a published third-party account of measurement of the amount of brown fat activity as a function of age on the left. And you can see how dramatically it decreases with age. To the point where you're over 60, you can see almost no detectable brown fat activity. What does this mean? This means this is an explanation for what we commonly observe as a consequence of aging. When we were young, if you were like me, you could totally pig out on ice cream and cake, and you wouldn't gain an ounce. We have, and as you get older, like I am, you barely go beyond your normal dietary intake, and you start accumulating fat, especially central fat, and most deadly is fat deposition in the wrong places in our body, which is, for instance, coronary disease. What the plaques in the coronary arteries is fat deposited in the wrong place. What causes this abnormality of fat metabolism? Well, we and the scientific community in general believe that a very important component is the loss of brown fat. Brown fat is a specialized tissue that is anti-fat. It takes sugar out of the blood and burns it to generate heat. And it also regulates numerous aspects of the body's metabolism. And its loss throws the balance of metabolism off balance. And so we start gaining weight, depositing fat in the wrong places, etc. So what would be the solution? The solution we're proposing is slide 21, is to transplant the brown fat tissue back to what you had when you were young. How would we get these cells? Well, pluripotent cells can make every cell type in the body, and we published results from our paper showing the generation of pluripotent stem cell derived brown adipocytes. And in slide 21, I'm showing you the purity of PureStem brown adipocytes. The marker here called UCP1 is staining the cells red. On the left is cells taken directly from brown fat tissue from a young person. And you can see the cells are there, but there's lots of impurities. On the right are our PureStem derived scalable pluripotent stem cell derived brown fat cells, and they're 100% pure. And they're young. And definitively brown fat, and we have filed patents on this manufacturing technique and the use of these cells in medicine. Slide 22 reminds us that the markets are very large for type 2 diabetes, which is caused, at least in part, by the loss of brown fat and the central obesity. And type 2 diabetes, it's estimated upwards of half of all Americans will have at least pre-diabetes in the coming decades, 2050. Large and growing markets, anticipated to be now well over $150 billion. And competing products, including those that we're talk about browning white fat, do not generate new brown fat tissue that can control metabolism through all of the facets that I previously alluded to. Vasc1. We chose products. We have over 200 potential products in our PureStem library. We chose to focus on brown adipocytes for the reason I just alluded to. Vascular progenitors, because vascular aging is the #1 killer in the United States, coronary disease, myocardial ischemia, and of course we have peripheral vascular diseases as well. We've similarly demonstrated that it's possible to manufacture 100% pure. You can see, for those of you who have a scientific bent, slide 23 on the right shows flow cytometry of these proprietary Vasc1 cells, and they're 100% pure as best as can be measured, and definitively young regenerative vascular endothelium capable of making new blood vessels, as you can see in the lower left. Vascular insufficiency and aging is an extremely large market and unmet needs. It's estimated that it's over $1 trillion market worldwide, and we consider that a very attractive market opportunity for this product. Lastly, I'll conclude with the most exciting technology I've ever been involved with. In my career, I've had the pleasure to be involved with the isolation and cloning of telomerase, which was an enzyme capable of immortalizing cells, led to Nobel prizes for some of my collaborators. I've enjoyed being involved with the first isolation of human pluripotent stem cells, and so on. I've never seen anything quite as exciting as induced tissue regeneration. I think it's going to take the world by storm. It's a really, really exciting thing. And let me try to explain in just a few minutes why. Slide 26, I'm showing you two pictures. On the left is an image that our scientists released back 10 years ago, in the year 2010. We demonstrated that it was possible to take cells from an aged patient and using certain, as we say here in the diagram, germline transcription factors, these are genes, we could reverse the aging of human cells in the laboratory dish, resetting the clock of cellular aging, known as the telomere clock of cell aging, and making the cells pluripotent again. So we transported skin fibroblasts from the skin back to these all-powerful stem cells from the very beginning of life. And, of course, that enables you to make any type of cell that's your own cell. It's genetically identical to you, but they're the young cells that you had when you were born. So you could replace them into the body with your own young cells that you had decades earlier. Pretty darned exciting. About as exciting as you could get, except for what we did next. On the right is the concept of cell aging reversal in vivo. What we did is we filed patents on a version of this technology applied not on cells in the dish, but on cells and tissues in the human body. As we say, an in vivo, or in life, in the body application, as opposed to in vitro, in the glass, or in the laboratory dish. Cell aging in vivo. And we call that induced tissue regeneration. Slide 27 shows a recent publication. Dr. Bailey mentioned a publication out of Harvard Medical School just in the last couple of weeks. The cover of Nature, a very prestigious journal, with the rather provocative title, Turning Back Time. Our competitors, really, at Harvard Medical School, David Sinclair's group, published their work in Nature on demonstrating that the technology that we've filed these early patents on is capable of reversing markers of aging in the mice and actually inducing regeneration. And their model was a crushed optic nerve, showing that the addition of these agents could take cells back, not back to pluripotency, not back to the very beginning of life, but part way back in time, reversing aging to the point early in our development when tissues are capable of regenerating themselves after damage, something that we do not have in adult humans. And so in this case, the optic nerve rewired itself. And believing that this would be the ultimate application of the technology is why we filed these patents, and we called the technology induced tissue regeneration. And it is the reason why we place such a high priority on it. It's commonly called, if you Google the term partial reprogramming, that's the term that the majority of scientists refer to it. Our in-house term is induced tissue regeneration. Slide 28, for those of you who want to follow the patent aspects of this to see who will have the important patents, I'm showing you now 4 published patent applications, some of which are beginning to issue around the world. As you can see in the top left, compositions and methods for induced tissue regeneration in mammalian species. And then on the right top, induced tissue regeneration using extracellular vesicles. This is a delivery modality patent. So we've been very aggressive at filing for intellectual property protection against unfair competition in this very important emerging area. Slide 29, milestones. So we plan to do more partnerships and licensing our UniverCyte technology, bailment of our master cell banks, and using PureStem derived biomaterials. We plan to complete the development of cardiac cells for drug screening, and as Dr. Bailey mentioned, an equity financing and Reverse Bio, currently a 100% owned subsidiary of AgeX, and the purpose of Reverse Bio will be to carry forward the development of ITR-related technology. Slide 30, in summary, AgeX is targeting some of the largest unmet needs in medicine in the United States, those of chronic degenerative diseases of aging. Again, 80% of healthcare costs are associated with chronic disease, and aging people have commonly 1 or even 2 or more chronic degenerative diseases. Those are what eat up the majority of healthcare costs in the United States. We'll be partnering some of the non-core commercial aspects of our technologies in pluripotency. We plan early and widespread commercialization through the bailment of our UniverCyte master cell banks, internally development of AgeX BAT1 and VASC1 and the cardiac cells for drug screening. And lastly, induced tissue regeneration for reprogramming aging, reversing aging in vivo, which will be developed by our subsidiary, Reverse Bioengineering, Inc. That concludes my presentation. As I mentioned, we'd be happy to hear from you. We enjoy interacting with you. Feel free to contact the company or to email me directly, mwest@AGEXINC, AgeXinc.com. I enjoy interacting with all of the stockholders, and we wish you and your families a very happy and healthy New Year in 2021. Thank you for joining us today.